Welcome back to What Did I Do in Lab Yesterday, where I tell you guys what I did in my biochem lab yesterday. Featuring results from my E. coli cloning labs. So after my regular lab section, I had to look at the bacteria plates from my last lab just to see what happened, and they went pretty well. So starting off, we have our control plates. There is no bacteria on here, which is there's not supposed to be, because this was just regular, changed bacteria. This is just a regular gel agar plate with ampicillin antibiotic in it, and the reason there's no bacteria on here is because we did not put the ampicillin resistance gene into this batch, so the antibiotic just killed all of the E. coli and didn't let them grow. That was supposed to happen. This plate has the bacteria with the ampicillin resistance gene, so that's why you see all of the growth on here. And it does have the digested plasmids that we made earlier, but it does not have any of the viral genome in it, which is why it is all blue. Whereas this one, you can see here, it has blue and white colonies, which means there is the bacteria that didn't pick up the viral genome, but there was also bacteria that did pick up the viral genome, but it doesn't have the dephosphorylated stuff, so there's a lot less white colonies than there is blue. This one has very few colonies and they're blue because there's only the digested and dephosphorylated plasmids, but not any of the viral DNA in it, so that's why there's only a few and that they're all blue. This plate, there's quite a few white colonies and only a couple blue colonies, which is because this one has the good dephosphorylated plasmids, which it makes it easier for the DNA to bind, and it also contains the viral DNA. And then lastly, this one is all blue because we didn't put any recombinant DNA in there, it's just bacteria in it. So there's no viral DNA, it's just here and it's just hanging out. So kind of what it means. Any white colonies mean that there is the new recombinant genes in there, so there is the viral DNA cloned in, and any blue colonies means that either there's no viral DNA there or the cloning wasn't successful. So that's just kind of what it means, but there's white colonies, which is great. That means at least part of it was successful.